The overall goal of this procedure is to enable high-resolution imaging of the poles of Drosophila egg chambers. This is accomplished by first dissecting ovarioles and staining them with fluorescent antibodies. The second step is to identify and separate egg chambers of interest. Next, selected samples are oriented and aligned, then embedded in glycerin jelly blocks. The final step is to cut and rotate blocks to mount egg chambers so the long axis is vertical. Ultimately, confocal microscopy is used to reveal cellular and subcellular organization at the poles of the egg chamber epithelium, such as border cell arrangements.
Existing methods to view specimens perpendicular to the imaging plane via optical sectioning and algorithmic 3D reconstruction can cause photobleaching and light scattering and provide poorer resolution, which yields less accurate images. The main advantage of this technique is that it allows us to mount specimens at a new angle and view them directly. We used this technique to image the poles of developing egg chambers, which provided insight into the organization of follicle cells.
Visual demonstration of this method is critical as the preparation and mounting steps are difficult to learn because they require micro-manipulation of stained specimens and reorientation of the samples from the conventional viewpoints.
Demonstrating the procedure will be Lathiena Manning, a graduate student in my laboratory.
To begin the experiment, obtain a glass depression slide and pipette several drops of dissection media into each cavity. Anesthetize the flies with